take your math benchmark today, people? You should have come in to do that. How'd you guys do in your math benchmark? Yeah, did you like doing it online, or would you rather have it on paper? Paper, online, you liked online? Online? Yeah. The biology benchmark tomorrow is online, but it doesn't have stuff you have to work out like on the math stuff. But they will give, did they give you a Scantron you also had to fill out on paper? Yeah, so I think this is the way they'll set up the biology benchmark. Again, the biology benchmark, I expect all of you to pass. Yes, what I expect more so is not just passing, I expect better than passing. Okay? The only unit we haven't fully covered is the one we're on right now. So you've got everything covered. And again, I'm going to take this data and we're going to look at it and see what kinds of tutorials do I need to have. Okay? So passing, as per the state, is a 42%, gentlemen. Let me say it again. Passing is a 42%. Holy crap, you better pass. Really? A 42? Yeah. I expect better than that. Don't you expect better than a 42? If you got a 42 on the test, wouldn't you just have heart failure if you got a 42 on a test? Yeah. Close. You better, Missy. If you got a 42 on a test, it better be bad. Okay, so you guys online, take a minute, set up your page. Remember, you can pause this whenever you need to, and let me fix the lights for you guys. Okay, this is Tuesday, yes? Tuesday? So 427, this is Unit 12, and that's on genetics. And this is period 7. So we started on our notes. We've done one page of the notes so far, and what we did is B6D, where we basically wanted to look at gene expression and realize that it's regulated. That's really it to it. I'm going to skip B6E and come back to that. What I want to do today is the start of B6F. It makes more sense to me to do this first. So I have a whole thick filled up page of notes. So what I decided to do is cut that in half because I don't want to take notes all frickin' period long. So that's kind of a bit much to take all at one time. So we're going to split this in half a little bit. So B6F says that you're going to predict possible outcomes of genetic combinations If I put combos, do you guys know that's combinations, right? So when you buy a combo meal at McDonald's or whatever, it's a combination of certain things, right? So you're going to predict the outcomes of genetic combinations such as, I need to move this up here a little bit, such as monohybrid crosses, which you've done before. These are your little Punnett squares, yes? You did Punnett squares with 4 boxes. Monohybrid crosses, that's exactly what they are. Dihybrid crosses. Crosses means, hey, we're going to make these two and look at the offspring. Okay? Dihybrid crosses and non-Mendelian inheritance. Non-Mendelian. Mendel is the father of modern genetics. So this is non-Mendelian inheritance. So I'm going to put a title on this one of just heredity. Okay, so a couple of things I want to point out here. And I'm going to grab a different pen so it shows up a little bit better. Mono, how many is mono? 1. So when we do a monohybrid cross, we're only going to look at one trait, like eye color, or whether or not you have dimples, or whether or not you have a widow's peak, or freckles or no freckles. We're looking at one trait. If we, that's the box will have, the Punnett square will have 4 boxes in it. If we look at a dihybrid, how many is di? 2. We're looking at 2 traits at a time. So what's the possibility of you having both a widow's peak and dimples? So it's like we hook them together. The box we have there is going to have the Punnett square we have there is going to have 16 boxes in it. So it's a bigger Punnett square. So you're taking into account 2 traits at a time. Okay? And so what's the chance of me having a kid with blue eyes and dimples? Yeah? So that's what you're looking at. So monohybrid only looks at 1 trait at a time. Dihybrid looks at 2 traits at a time. And then this non-Mendelian stuff means it's not following the laws of genetics that Gregor Mendel came up with. So we're going to start off here, and let's start off with the word genetics here. So if you get a little simple definition of genetics, kind of a generic definition, it says that it's the study of heredity. That's a little bit too simple. And you guys should know that when we talk about heredity, you're talking about passing things on from parent to offspring. Right? Whether it's money or stuff you inherit from your parents, your grandparents, whatever, passing them on from parent to offspring, right? We can talk about it in biology, but when we talk about heredity in biology, it's all about genes, all about DNA stuff. So this is a nice kind of baby definition, genetics, the study of heredity, but we really, when we talk about genetics, we're talking about 2 distinct things here. So one, we're talking about how physical traits are coded for. And expressed. So how physical traits are coded for and expressed. All right, so here's the deal. Coded for means it's the DNA. More specifically, the nitrogenous bases, right? What are the nitrogenous bases? Yeah. So that's one part of genetics. We just spent a lot of time working at that, right? And then expressed, how they're expressed, we're talking about transcription, and what's the other one? Translation. Yeah. So our whole unit 11 was talking about genetics too. It's the first part of genetics. How are the physical traits coded for? That's the DNA. It's in the order of the nitrogenous bases, right? What's the sequence or the order of those bases? And then how is it expressed? It expresses itself by making proteins through that process of transcription and translation. Yeah? So that's the first part of what genetics is. The next part, number 2, which is also something that we talk about with genetics, is how the traits are passed on. So we have 2 parts. So it's that how aspect. We also talk about probabilities. Like what's our probability? What are the chances of having a kid with the blue eyes and the dimples, or the widow's peak and freckles? We talk about probabilities. So how traits are passed on, and I'm going to put in parentheses, and probabilities. And that's when we use those Punnett squares to see, well, what are the chances of me having a kid like this or a kid like this and so on. We keep it simple and only do like 1 or maybe 2 traits at a time, because in reality, how many traits do you have? Yeah. So if we tried to do all of those at one time, I mean, if we only did 3 traits at a time, our Punnett square would have 64 boxes. That's only 3 traits. Let's say eye color, hair color, and whether or not you have dimples. That'd be a 64 square Punnett, 64 box Punnett square, right? Can you imagine trying to fill that out? It gets really complex. And 3 traits is not enough. Tons of traits, right? So when we do these Punnett squares, what we do is we try to keep it really simple and clean, straightforward. So we get the general process of it. And if you decide to take genetics classes later on, and you can get into a lot more detail than that and figure things out. And they also have computer programs now where they can plug things in and it'll do the statistics, the probability Punnett square for them. So that makes it a little bit more reasonable there too. But it's just talking about, let's include a whole bunch of traits at one time here. Okay, so the next one we're looking at, we've got the basic idea of what genetics is. The next thing we want to look at is the other part. This talked about non-Mendelian inheritance. We need to know what Mendelian is before we can talk about, well, what's not that. So the next piece we're going to do is we're going to talk about Gregor Mendel. I'm not going to test you on dates, but I always like to know when in time this happened. He did his work in the 1850s. So no electricity, right? No indoor plumbing. Yeah. He was actually an Austrian from Austria, an Austrian monk. And he worked with pea plants. That's what his specimens were. Peas, you know, like peas that you eat? That's what he worked with. And so all his laws that he came up with, which are still true today, he did this by working with pea plants. So they would have greenhouses back then. They still had glass back then too, so they could have greenhouses. And I'm sure he probably had to have something because he hand-pollinated everything. He didn't let the bees and everything pollinate. He would take the pollen from one flower and carry it across and say, I want this plant to pollinate that plant right there, so he could control what everything did. And so that's what he did there. And can you imagine having to work like that? You've got, 50 pea plants and you're keeping track of everything and saying this plant's going to mate with that one, and just, yeah. Statistically, keeping up with all that stuff, and it's not like they had paper like this, right? It was some serious work to do stuff like that. And in the 1850s, what's sad about him is he died thinking that people didn't think his work was any good. Because, they used to have these big, every few years they'd have these big gatherings, I suppose you could say, of scientists, and they would kind of present material that they'd been studying on. And his was kind of shoved over to the side like, you don't know what you're talking about. And it wasn't until about 20 years after he died or so, and somebody was going through all of the papers that were at the monastery, and they found his work and thought, holy crap, this stuff is good, it's really true. And so it wasn't until after he died that he realized, yeah, he got that fame from that. Every single biology book you pick up is going to have his name in it. I don't care what country it is, what language it's in, this guy's name is in there, Gregor Mendel. He's considered the father of genetics. The father of genetics. And he came up with two laws that are still useful today, and they've tweaked them a little bit, but remember now, he's in the 1850s, he didn't know anything about DNA, he didn't know anything about chromosomes or stuff like that. He was just looking really at traits. He didn't even use the word genes. We're going to use the word genes in this, but he didn't even use that. He used traits. Your traits come in pairs. So he had two laws. Here's his first law. Okay, so the first law says that genes for each trait come in pairs. Okay, I'm going to put a little box around this and put a little asterisk here. Because when I say pairs, what we understand today is a little bit different from what he got. We can expand it today. So when we talk about eye color, we say you have two genes. One might say brown, the other might say blue, and the brown's dominant, the blue's recessive, yeah? Okay, you guys are familiar with that. In reality, it's not that you just have two genes, because if that were the case, if you had brown eyes, every single pair of brown eyes in here in the school would be exactly the same brown, right? Or every single pair of blue eyes would be the exact same. Are they? No, there are all sorts of shades in there. That leads us to believe that there's not just one pair of genes that says eye color, there's probably a group of them. Now, they're all together, right? So we've got them all in the same chromosome. They're all together. They stay together. So they kind of go as a group here. But I'm going to drop down here, and I'm going to put towards the bottom of the paper, I'm going to get my asterisk and put it in here and say, or multiples of two. And a lot of traits are that way. Now, the reason why we don't spend time much on that is because it makes it a lot more difficult to explain if we keep saying, or multiples of two. I want you to be aware that you really do have more than two genes for each trait, for most of them, but they're always in pairs and they're always linked together. So you're not going to have eye color on two different chromosomes. It's going to be on the same chromosome, right? So all the stuff for your eye color is going to be on whatever chromosome it's on, and that's where it's going to be here. So this, if we just talk about them coming in pairs, then it makes more sense, and we can do simple things like Punnett squares. You just understand that, yeah, it's a lot more complex than that, right? Yes? Okay. So his first law says that the genes for each trait come in pairs that separate. Oh, sorry. That separate during meiosis. What do we make during meiosis? You remember? Body cells or sex cells? Sex cells during meiosis. Remember, meiosis is going to make so that each sex cell, or how about saying this, during meiosis, so the sex cells only have one gene each. Remember, sex cells, otherwise known as what? Gametes. How about 4 each trait? There, that makes more sense to me. Okay, there's this first law. So when you have these genes, one of those genes, if you've got 2 genes for every trait, one gene goes into one sex cell, one gene goes in the other. Yeah? That's what he's saying. And when we look at our paper that we were working on with the karyotype, then what we have here is what we're talking about. So here, when they made sex cells, one of the chromosomes went into each one. So we have 2 chromosomes for everything, right? Our chromosomes come in pairs, our genes come in pairs. And the number 1 pair, one of them goes here, one of them goes over there. They're going to be separated from each other. So this is the number of chromosomes in a sex cell. It's half the number, right, that you should have. So when one sex cell fertilizes the other sex cell, then you have the full complement, the total number of stuff that we go on. So that's what he's saying. He figured that out. He's the one that figured out, hey, your traits come in pairs, and when you make sex cells, so half of them go over here and half of them go over here. So that now here's the mom, here's another dad over here, and when those get together, you have the right number. So you're picking up traits from both parents, aren't you? That's a key. They didn't believe that. They thought, okay, there are some really weird things, but at the time they thought that the sperm had a perfectly little formed human in it. Nothing to do with the mom. It was all the man. Had a perfectly formed little human in it, and the mom was just basically an oven. Just cook it up. And that was about it. That was one of the really common theories of the day. So what he's saying here is really different compared to what was believed at the time. Yeah, I'm sure they did too, but yeah. But the scientific explanation, that wasn't it. Okay, his second law says different genes separate independently from each other. And I'm going to put a little caveat here, a little extra something here, unless they're on the same chromosome. But he didn't know about chromosomes, remember? So for your eye color, then they're all probably going to be on the same chromosome, right? So there are some genes that are kind of linked together just because they happen to be on the same chromosome. You either get this chromosome or that chromosome. So some things are linked together. So 2 things. He said, hey, your genes come in pairs, and when you make sex cells, each of the sex cells has one of those genes. And that, the second law, genes separate independently from each other. So chromosome number 1, you have 2 of those, right? It doesn't matter which sperm cell or egg cell it goes into. Yeah. Chromosome number 2, it doesn't matter which sperm cell it goes into. It's going to have one of them. Chromosome number 3, the pair of them, one of them is going to go in. It doesn't matter which one. the first ones of each don't have to go into one sperm cell, and the second ones of each have to. It doesn't work like that at all. Okay? So they separate independently of the other. This is otherwise known as, put a box around that one, independent assortment. That's huge because that increases diversity. Independent assortment. So that's what we talked about with your chromosomes that we were doing yesterday. They separate independently from each other. So again, the chromosomes, they don't have to stay together. As long as you have one of each of them in the sex cells, it doesn't matter whether it was the one from your mom or the one from your dad. So independent assortment is what we have here. Okay, once you've written that down, flip over to page 3 in your packet. Okay, and I'm looking at the first karyotype that we have here. And this karyotype, now typically the only time we're going to see chromosomes is when it's going through mitosis, and so we have too many chromosomes at that time. We have 92. How many are you supposed to have? How many chromosomes are supposed to be in each of your human cells? How many? 46, and then they come in pairs, so we say you have 46 chromosomes or 23 pairs, right? Now, this one has 23 pairs. This is called diploid because you have 2 chromosomes for each one. You don't just have one of the number ones, you have 2, so it's called diploid. If we look at a sex cell, and I'm going back over here to page 39, somewhere in there. If we look at a sex cell, here, notice there's only one of each chromosome, and this is called haploid. Haploid means you have half the number you're supposed to have. That's what the sex cells are. So your body cells are diploid. They have the full complement, 2 of each chromosome. Your sex cells are haploid. They only have half. And that should make sense to you because you get half when the sperm fertilizes the egg. There's half. The egg has half, right? Okay. Now, the reason when you looked on this page that grandma and grandpa's karyotype or mom and dad's karyotype have 4 is because we got this karyotype, we got it from a cell going through mitosis where they had double the number. They had double everything. Because remember, in mitosis, you're making a new cell. So you take the chromosomes, you make copies of everything, and then you split them equally into 2 new cells. So now you started out with 1 cell, you have 2, right? So it makes sense that we have to have extra of everything, yeah? So this, they're just showing you that, hey, this has 92 chromosomes because it's about to split into 2 new cells. So when they do a karyotype, they actually get cells that are going through mitosis, and then they have to trash half of the chromosomes because there are too many of them in there because they haven't split. They keep the cell from going the rest of the way through mitosis and splitting. Okay, flip over to page... Yes. Yeah, they just ran. As long as they have one of the number 1s, one of the number 2s, I mean, it's specific in that sense. Yep, close, but it has to be one of the number 1 chromosomes, one of the number 2. Otherwise, it's bad. You know, remember that one, if we had the karyotype where they had 3 of the number 21s, that's bad. There would have been a sperm cell or an egg cell that had 0 21s. So, you know, apparently those are the ones that just don't even live at all. So if you have 0 21 chromosomes, then it doesn't even survive. Oh, and I need to turn this off, don't I?